So I'd like to I'd like to start by thanking the Kavli Foundation for sponsoring this meeting, and for the Kavli Institute for handling all the logistics so well. And thank you all for coming, and especially thanks to the students and postdocs who are willing to present posters in this new and rapidly emerging field, and kind of being the first penguins willing to be shoved into the waterhole. But don't worry, I don't think anyone's going to be particularly fierce at this meeting. It's a very welcoming community. So what I'm going to tell you about is how we can connect our dynamic gut microbiomes to the brain. And so in the first part, I'm going to show you some results that show just how dynamic and plastic our microbiomes are. And then towards the end, I'm going to connect them to the brain and behavior. And this is going to be primarily focused on DNA sequencing. Peter Dorrestein is going to be speaking a couple of talks down and tell you about the metabolomic side of this equation, which is also very exciting. So in thinking about our bodies, naturally we initially think about ourselves as having genomes that are complicated, that make up, that provide the instructions for producing our bodies. But amazingly, to the 20,000 human genes that each of us carries around with us, we have 2 to 20 million microbial genes in microbial genomes on and inside our bodies. And that's truly amazing, right? Because what that means is that at the genetic level, in terms of unique genes, we're only about 1% human. And despite all the excitement about systems biology, systems medicine, and so forth, it's pretty difficult to do systems anything if you're ignoring 99% of that system, which is essentially what we're doing when we neglect the microbial sides of ourselves. And what's particularly remarkable is that unlike our human genomes, which we're born with and we can't really alter, with our microbiomes, we're changing them every day of our lives. And just imagine the potential if 99% of your genes could be added or lost based on things that you did and could potentially take control of if you knew how. So in the Human Microbiome Project, which I was involved in several capacities, we set out to characterize a baseline human microbiome that you can use as a resource to add your own microbiome data to. And so this was a large NIH-funded project, a total of $173 million. And the largest single component was a study looking at about 250 people at up to 18 sites in the body. And in this project, we collected 4.5 trillion bases of DNA, so 1,500 human genome equivalents, which was a lot for something published in 2012. And so the great thing about this is that we had a tremendous amount of sequence data, but that was also the terrible thing about the project. And just to illustrate this problem, I'm just going to show you the first file of data from the Human Microbiome Project. And so despite the fact that what we're fundamentally doing is ecology, it's pretty hard to tell who lives where in the environment from this sort of data, right? Like you probably can't even tell that's an oral sample, let alone the sequence signatures that let us find that out. And this is especially a problem today because you have people coming into their physician's office with a readout of their microbiome that they got either from projects like the American Gut Project that I run here, which is an open source crowdfunded project, or various companies where they come in and they say, hey doc, I have some great news for you, which is that I had my microbiome done. Now I have this list of 1,000 species that are in my gut. And with all this data, you can tell me what's wrong with me, right? And I mean, what's your physician going to do? They can refer you to colleagues in psychiatry for being crazy enough to think that it's going to help, right? But I'd argue that our mission at the moment is to make it not crazy anymore and figure out how to use these massive data resources that we have available, things like what's going to be available through the Data Science Initiative, and what's already available at SDSC and PRL and so forth, to take those individual profiles or a time series of the profiles and turn all that sequence data into something you can interpret and use, so essentially providing a user interface to your microbiome. So when we apply tools we developed in my lab to the microbiome, so this in technical terms is a unifrac distance matrix that's been reduced into a principal coordinates analysis plot. And so what we're doing is we're exploiting the evolutionary distance between different microbes to place the whole communities on a map. So each point on this map represents all the complexity of a given microbiome, boiled down to just one point after we read it out from its DNA. And 2 points are close together on the map if they have microbes that are more similar in terms of their evolutionary history. And they're further apart if they have microbes that are more dissimilar in terms of their evolutionary history. Now, when you think about those 250 people, you could think of all the things that could make a difference to their microbiome, like how old they are, whether they're male or female, how much they weigh, at what time of day the sample was collected. All kinds of things could affect the location of each microbiome on that map. And when we color by the main factor, it's immediately obvious what's driving the system. So the different parts of the body have really different microbiomes. So the mouth in green is totally different from the skin, from the vaginal community, from the fecal community. And to highlight this, I'm just going to highlight the first person, their mouth and their gut sample, first person in the HMP at the first time point. And they're in totally different places on this map. And you might be thinking, well, so what? So what does that really mean? And it wasn't until we did the Earth Microbiome Project, where we looked at tens of thousands of environmental samples, that we could really understand what these profound differences in the microbiome meant, because we could go out on the planet's surface and ask what two samples are just as dissimilar as the mouth and the gut of the same person. So if you think of your mouth as being kind of like a coral reef, you have these complex mineralized structures covered with biofilms that perhaps your dentist pesters you about from time to time. The amazing fact is that your mouth is as far away from your gut in terms of its microbial ecology as the microbes in the water of this reef are from the microbes in the dirt in this prairie. So they're essentially non-overlapping microbiomes. And that's amazing because what it means is a few feet along the length of your body makes as much of a difference to your microbes as thousands of miles across the Earth's surface. And so you want to think of yourself not as having one microbiome, but as having a whole archipelago of different microbiomes throughout the body. And we're just starting to understand the connections between them. So you might be wondering, well, how do you travel through this microbiome map from birth? And this is work we did with Ruth Lay, and this visualization was done by Antonio Gonzalez and Yoshiki Vasquez, two very talented computer scientists in my lab. And what we did is we inserted the developmental trajectory of this one infant into this microbiome map provided by the HMP. So we're starting off with the infant's first fecal sample, the meconium, which shows up in the vaginal region of the plot. And amazingly, it turns out to be true that your first microbes are determined by your delivery mode. And as you pass through the birth canal, you get a microbiome all over your body, including your gut, that resembles the vaginal microbiome. And so what I'm going to do is I'm just going to connect up the trajectory one time point to the next and ask how much it develops towards the adult fecal state over the first 2.5 years of life, where most of development, including a tremendous amount of brain development, is going on. So the question is, how completely does it approach the adult state? Is it fast or slow? Is it a smooth progression, or do things happen along the way? And in case you're wondering why 2.5 years, that's when the kid was toilet trained. And as you can imagine, it's a hell of a lot easier to get the sample out of diapers than it is to fight a toddler who's probably learned how to flush for every single sample. So a lot of these studies tend to end at about that age. So anyway, I'm just going to start this going, and what you can see is sometimes there's a lot of change 1 week to the next. Remember, each time point's a week. Others, there's a little change. And remember that what matters on this map is how far apart two samples are primarily. And the distance a child travels 1 week to the next is larger in some cases than the distance between two different people in the HMP. So if you're thinking that your kid is a different person 1 week to the next, that can literally be true in terms of his microbiome, which remember is where most of his genes are anyway. So coming up here is something fascinating. So he gets antibiotics for an ear infection. You see that massive regression of the microbiome followed by recovery. And that went by pretty fast, so I'm just going to rewind it for you and play it for you again. What you can see is that on administration of oral amoxicillin for an ear infection, we see this massive regression in the microbiome, almost perfectly undoing months of normal development. And then in this case, he's incredibly resilient, so he gets back to where he was initially. And by 2.5 years, he's basically in the healthy adult state, which is what we also see when we compare kids cross-sectionally. But that resilience isn't true for every child, and a main focus of our work at the moment is trying to predict from the microbiome, are you going to be resilient to antibiotics, or are you going to go into a different microbiome state from which you might not ever recover? So a lot of people have been interested in the possibility of placing themselves on this kind of microbiome map. And this is Larry Smarr, who's our director of CalIT2, and shortly after I arrived, he took me over to his display wall that's about a 10-minute walk from where we're walking, where it's a 64 million pixel wall, about the same size as that wall, and a high-resolution data display. And I was taking him through the Human Microbiome Project data, like I just showed you, and he said, well, you know, Rob, I have a very interesting microbiome myself because I'm an IBD patient, and how do I get myself onto that map? So I moved to UCSD just a couple of years ago, and as you know, when you move institutions, you expect a certain amount of crap from your colleagues. And in my case, it's literally been true because this box, I don't really need to say any more about that. But Larry's a leader in the quantified self movement, and he started by measuring his own weight back in 2000, so one dimension, and then he's added additional, like dozens of dimensions with blood tests, thousands of dimensions with SNP profiling, and now millions of dimensions with the microbiome to this data display. And for a period there, he was already collecting his own stool. And when you look at it on one of these taxonomy plots, so the different colors just represent different kinds of microbes over time, if you're looking at it, you probably don't get a lot out of that. And that's okay, because Larry didn't either. And I hate to think how much all the supercomputer time and personnel effort that led to this graph must have cost. But when we reanalyze it with our improved visualization tools, so exactly the same data, this was put together by Justine Debelius, who ran the Robert Wood Johnson Foundation symposium we did last year, which was very successful. What you see is this much clearer pattern where he starts off at one edge of this blue region, switches over into the red, and then bounces around at random in the red region. Exactly the same data, much clearer interpretation. So you're probably thinking, well, what does this actually have to do with Larry's health? And so because Larry collects all this clinical data, we can actually match them up together. And so what we see is that this initial shift here is caused by antibiotics. And then we see he's in this period where his microbiome is changing directionally. He has frequent IBD symptoms and his life is not going so well. Changes his medications, which switches him from the blue into the red. And then in the red region, where his microbiome is just bouncing around at random, his weight goes back up to a healthy set point, and then it stays there. And had we known about Larry's individual microbiome states, we could have told him as soon as he got into this red region that he was going to get back up to a healthy set point and recover. And so this is why we critically need to understand microbiome dynamics, both at a population level and at an individual level, to start to understand what states are healthy for people and go beyond description and towards prediction. So one really important question that comes up in all this is whether controlling single pathogen species has led to a kind of microbial silent spring. And Janelle did a beautiful job of showing you the effects of individual pathogens on behavior. But when we consider the whole microbiome, as everything from measles to TB has been brought under control, you have this explosion of autoimmune diseases, everything from Crohn's to diabetes to asthma to MS that have skyrocketed over that period. And so one possibility is that we may be missing microbes, and Marty Blaser has written a wonderful book exploring this at greater length, that we may have got rid of microbes that we've co-evolved with, that we may need to resupply to be able to stay healthy, just the same way that a century ago industrial processing had stripped a lot of naturally occurring nutrients out of food that have to be resupplied, like everything from iodized salt to vitamin B supplementation in bread. And so to study this, we've been turning to some of the most most traditional living people there are, like the Yanomami, pictured here in the Amazon basin, and the Hadza in Tanzania. So in work with Maria Gloria Dominguez-Bello, we've been able to show that they have far more diverse microbiomes, that they are completely different even compared to other non-Western populations that have entire phyla in their guts, like spirochetes, that I guarantee no one in this room has in the gut, and then tremendous differences in a whole range of different taxa. So you might be wondering, well, what does all this actually have to do with the brain? And we're seeing a lot of connections. So one thing you might have read about last year that we did with Chris Lowry's group at Boulder was that you can actually vaccinate mice against stress and anxiety by exposing them to Mycobacterium vaccae, which is a bacterium out of the soil. And there's a lot of links that are starting to come together, where essentially we have systematic communication between the brain and the gut, both neurally and then systemically through neurotransmitters and cytokines and so forth. And so this concept of the microbiome gut-brain axis is starting to be untangled in mechanistic detail, as you heard from Janelle, and as you'll hear in far more detail from Sarkis when he gives his keynote talk later on. So I won't get too much into this, except just to note that there's a whole range of diseases that are now being linked to the microbiome, where we had no idea it was involved in them just a decade ago. And the biggest way you can change your microbiome is through diet. So if you're a mouse and you change the diet, one day after you do that diet change, you have a completely different microbiome. This is work we did with Jake Lucis, looking at 50 genotypes of mice on high fat in blue versus low fat in red diets. You can see that all the variability is explained by diet on this microbiome map. There's essentially no effective genotype. In work with Gary Wu and Jim Lewis and others, we also showed that this was true in humans, where if you eat, for example, a lot of carbohydrates, you have a lot of Prevotella. If you eat a lot of protein, you have a lot of Bacteroides, and other specific associations between long-term diet and the microbiome. We got excited about this and tried to get people in an inpatient setting to modify their microbiome experimentally over 10 days. And the results of that were very disappointing. So you can see that it just clusters by what person it is and not by which diet they were on, whether it was high fat or low fat. So what you eat today has very little effect, but what you eat over what you ate over the last 6 months to a year has a huge impact on your microbiome. And you might have seen some articles claiming otherwise, but it's very important to check exactly what they're claiming, because if you look at what's going on in this plot, so this is some very elegant work by Pete Turnbau's lab, what you can see on the plot is a measure of variability and how that variability changes when you switch people from a more or less regular diet to, in this case, a vegetarian diet. Rachel Dutton, who's now a faculty member here, is one of the co-authors of this paper. So this is really cool in terms of showing how the variability in your microbiome changes in a way that's reproducible between subjects. But if you look at the actual pattern of change, if you look at an individual subject's trace, so this is the same subject on low fat versus high fat, where you can see that the all meat, egg and cheese diet has a lot more variability. And then this is also true, that's also true for a second subject who's going to fill in over here, where you see a lot more variability. But if you put all the subjects on the same map and you ask, well, what does the variation for each subject look like? What you see is that, again, basically all of the points cluster together for each person, and it's very difficult to make yourself look like another person simply by changing your diet over a few days. You really have to be in it for the long haul. So this sort of result has inspired a lot of people to try to find a pin for themselves on this kind of microbiome map. And so to enable this, we started the American Gut Project, which we launched over Thanksgiving in 2012, Thanksgiving being a time when a lot of Americans are thinking about their gut for some reason. And then recently moved to UCSD, where we use crowdsourcing and crowdfunding for people to place themselves on the map. And this has been remarkably successful. So at this point, we've raised over $2 million, processed over 12,000 samples, and made all the data freely available in advance of publication in this open source effort. And on the scale, we see all kinds of cool associations between the microbiome and things you might not have expected it was involved with. So you probably guessed that your microbiome changes as you age, which it does. But another thing that's just as significant is how many hours of sleep you get at night. And you might not have expected that the microbiome was involved in that, although as Sachin is going to tell you later this afternoon, there's tremendous involvement there and very elegant work to understand the mechanism. And in terms of what affects your gut microbiome, these are power curves put together by Justine, who I mentioned earlier in connection with Larry's samples. And basically the x-axis is the number of people per group you need to see an effect. And the y-axis is the statistical power of the test, so 80% to 100% power to detect is up in this range. And so you can see that age and IBD and antibiotics, you need a few dozen people per group to see those differences. But the thing that has more effect than anything else with the steepest curve is how many different kinds of plants you eat. And we never expected that. But we also see all kinds of other things, like whether you're male or female, how much sleep you get, how fat you are, how much you drink, even how much you exercise, or I should say how much you say you exercise, because all this is self-reported. So we either see an effect of exercise or an effect of lying about it, and we're working with UCSD Student Athletics to try to resolve that question at the moment. Okay, so diet might affect the brain by these profound changes in the microbiome, but you might be wondering, well, how else can we do it? So one way we can do it, and Sarkis is going to really focus on this, is by activating the immune system in different ways and doing transplants into germ-free mice. And you'll see this stuff later on. And as you had David Brenner mention in the introduction, this really changed how we think about psychiatric diseases like autism in connection with the microbiome. Another thing you can do is immunization. And so I mentioned briefly the work we've done with Chris Lowry, looking at Mycobacterium vaccae and stress resilience. And so what's amazing about this is that when you inoculate mice with M. vaccae, and then subject them to this chronic stress condition, afterwards you see far more displays of proactive behaviours and far more resilience against the stress. And we also see that coupled to changes not just in their static microbiome state, but in how the microbiome itself changes over time. There's also been a lot of work on probiotics, and so again, I'm just going to summarize this really briefly. But there's a whole range of probiotics that have been shown to have psychoactive effects. Although at the moment, essentially all of this is in mouse studies. And in a commentary on Sarkis's paper, we were really able to outline this framework for how we could develop additional, more effective probiotics than the traditional ones that are so generally recognized as safe and come from fermented foods and so forth. Where basically the idea is that in the lab you can look for differences between healthy and diseased mice, either in terms of the microbes or the metabolites, or ideally both. And then you can take your unhealthy mouse model, try to remediate it in different ways, and shift the microbiome back into the right state. And then in your clinical trials, the idea is that you could try an equivalent modification of the microbiome in the human subject. And so there's evidence that you can do this kind of thing now for diet, for probiotics, for prebiotics, for antibiotics, and even for fecal transplantation, although you have to remember that that requires IND. And just to give you an idea of how dramatically fecal transplantation can reshape your microbiome, this is just to reorient you on this microbiome map. And I'm going to show you some microbiomes from C. diff patients, where you can see that their fecal microbiomes look nothing like, so there's these orange spikes that look nothing like regular feces. And so what's going to happen, and this is work we did with Mike Sadowski and Alex Karutz, 4 of these patients are going to get fecal transplant from one donor. And if you're wondering about how we do the fecal transplant, this is Bill Sanborn, chair of GI, about to administer one using hospital-grade stool from a nonprofit called OpenBiome. And so anyway, so what's going to happen is 4 of these patients are going to get a fecal transplant, but instead of each frame being a week, like I showed you in that infant, each frame is going to be just 1 day in the lives of these subjects who are experiencing several dozen diarrheal events a day and have failed antibiotic therapy for 2 years to be eligible for this trial. And so the question is like with the infant, like how much will it come to resemble the adult state? Is it fast or slow? Is it smooth or what? And I'm just going to start this going, and what you see is immediately, so in just 2 days, their microbiomes entirely reshape themselves into the healthy state, and then they stay there for months. And during that 2-day period, all their clinical symptoms are gone. And in fact, the last large-scale trial directly comparing antibiotics to C. diff in a clinical trial setting had to be stopped early because the antibiotics were about 30% effective, which is typical. The fecal transplant was over 90% effective, which is also typical. And it was considered unethical to continue withholding the fecal transplant from the people who were on the antibiotic arm of the trial. So they all got the fecal transplant, and essentially all of them recovered. And so the challenge facing us today is to figure out for what other disorders can we reshape the microbiome back into health, whether it's by something as extreme sounding as fecal transplant or benign sounding as diet or any one of those other interventional strategies. And so there's a whole range of diseases that are coming up as linked to the microbiome that we may be able to do this for. And in this respect, like a lot of people gave me flack for putting autism on this graph. But a lot of what we have to do is we have to find the good and the bad places on the map. And in fact, in the case of autism, a consortium of 3 groups from Arizona earlier this year showed that they actually got improvement of GI and cognitive symptoms in an open label study of doing fecal transplant for autism. So there's some problems with this. It's open label, it's only 18 kids, it's only for 8 weeks, but at the same time it provided a remarkable improvement on both ends of the gut-brain axis and clearly shows that more follow-up on this kind of thing is needed. So our dream with this, though, like at the moment the microbiome sequencing is slow, and if you send your kit into American Gut, it might take weeks, or sometimes if we run out of reagents or something, or the sequence is down, even months to get your result. And our dream is to move this into something you can do in your home and make characterizing your microbiome as natural as breathing into a mirror. So the concept is that as you examine yourself in the mirror, perhaps as you breathe on it, the chemicals in your breath are going to be whisked away into a mass spec and give you some sort of readout directly on the mirror as a display screen, presumably in some sort of more user-friendly format than this, but we're working on it. So then the idea is to use the same kind of machine learning that backs Google Translate to translate those metabolome profiles into microbiome profiles like you would get from American gut, and then place yourself on that kind of map. So perhaps telling you if you have a particular risk, what you should do in order to mitigate that particular risk based on data ideally from human clinical trials or perhaps from model animal trials. And then the idea is that we could even go a step further and have that display screen communicate. For example, you might have a smart toilet that does a direct readout of your microbiome. You might have a smartphone where during the day when you're in the supermarket and you're faced with a giant wall of yogurts and you need to figure out which one to pick for the probiotic you're looking for, maybe it'll use augmented reality so you can pick the right one off the shelf, scan the barcode, and figure out that you got the right one. So this probably sounds like absurd science fiction, but on the other hand, if you consider the distance between the Cray 2 and your smartphone, the 30 years of that advance in computation has been packed into just 10 years in DNA sequencing. So we can expect a comparable advance in the next 10 years that's going to separate the large sequencing instruments we have in the lab to what you're going to have that's handheld and battery operated. And so we need to start thinking about this now. And most of the pieces of technology to do this already exist. It's mostly an integration task. And so to do this sort of thing is exactly why I founded the Center for Microbiome Innovation last year, where already we have over 100 faculty from all over campus coming together to try to build the next generation of these technologies in collaboration with an expanding consortium of companies, both on the technology end and on the applications end. So with that, I'd like to thank the very large number of people who've contributed to the work that you've seen here, many of whom I mentioned during the talk, but I certainly can't read out this list of people in my lab who it's been a true privilege to work with. I guess I accidentally deleted the slide of collaborators and funding agencies, so apologies to them for that. But again, I mentioned much of that through the talk. And with that, thanks again for coming. I'd be delighted to answer any questions that you have. Thank you.
Time for a few questions. Nick?
I got the impression from your remarks that most of the disorders derive from the fecal family and that the oral and skin and vaginal families are less damaging. Is that correct?
Well, that's a great question, and I see Eric making for the door before I put him on the spot. So we've linked the oral microbiome to migraines. And with a reasonably good mechanistic story for how that might happen. And then on top of that, as a biomarker, the oral microbiome has been correlated with rheumatoid arthritis, with hepatitis, with diabetes. Let's see, I think that was type 2 diabetes, and then with a number of other conditions. So the reason we know so much about the gut is that a lot of this started with gastroenterologists. I think we're going to find more and more fascinating connections with the oral microbiome, which also has high biomass and ability to cause physiological alterations. With the skin microbiome, so for example, Rich Gallo has shown that amazingly whether or not you get sunburn from the same dose of radiation depends a lot on your TLR activation, which is largely determined by your microbiome, and all kinds of other things. So we know more about the gut right now, but I think we're going to see emerging roles for a lot of these other microbiomes as well.
Yes.
How do you track when you say diet, either the type of food that you're eating, or say with cabbage, raw versus cooked or fermented in the range?
Yeah, that's a great question. So so the FFQs only capture type, not cooking method. We've done some controlled experiments on cooking methods for potatoes with Jaron Vandermeiler at Penn State, Peter Dorrestein here, basically taking the same types of potatoes, cooking them in different ways and running them through the mass spec to see what's in them. And both so horrifyingly, fries are America's #1 source of vitamin C. Like there's not a lot of vitamin C left in each fry after you fry it, but if you eat a lot of them, which people do, then it adds up. So both for beneficial compounds like vitamin C and harmful compounds like acrylamide, both how you cook it and what the input ingredient is are critically important. So we're actually in the process of launching a food project that I think Peter is going to talk about, so I'm not going to say too much about it here, that's going to complement American Gut with what goes goes in the other end, and really start to understand all of these kinds of interactions. The other thing is that there's all sorts of compounds in food that aren't supposed to be there, like, say, antibiotics and antifungals in illegal fish farming. So even if you think you've never been on antibiotics in the last year, that may not be true, and we can find out just by looking at your fecal material, either microbiologically or chemically. So again, it's a very exciting area. We're doing some specific work on fermented foods with the Fermentation Festival of San Diego. So I spoke there 2 years ago, and then this past year, Rachel and Embriette and I all gave shorter presentations. And so scaling up that relationship is something that we want to do via the CMI. But there's, again, a lot of excitement.
All right, thank you.
One more question?
Just one more.
Yep.
Did you say where to get that mirror mass spec at? Because I can't find it on Amazon, so.
As I said, we're still working on it. But you know, I think if someone asked you about, say, in 1985 about your car phone and like, could you stick a 1-ton liquid-cooled supercomputer in the back of your car, and then could you couple them together, and then could you ship 1 billion units of it worldwide and network them all together, you probably would have thought that was pretty implausible as well. And when I was at TED in 2014, Ray Kurzweil gave this awesome talk about, like, all of the things that he said, and then the audience laughing at him for saying them, and then, you know, a snapshot of 10, 15, 20 years later, you know, yes, you really do have a computer in your hand, and it talks to other computers. So, so I wouldn't necessarily extrapolate from the fact that it's not available now to the idea that it'll never be available.
All right. Oh. You know, we're going to have a reception after this, and you'll have an opportunity to come up to the speakers and ask for additional questions at that time. Thanks, Rob.
Thanks.
Thank you very much, Rob.